Hi, thank you for coming. So it's my pleasure to introduce our seminar speaker today, Dr. David Weiss. Dr. Weiss is the founding director of the Antibiotic Resistance Center here at Emory. He first started his academic career by getting a PhD in microbiology from New York University. He then went and got his postdoc from Stanford University, where he had a postdoctoral fellowship from the Guiani Family Foundation. He has been at Emory since 2008 and has won numerous awards, including a Burroughs Wellcome Fund Investigator in the area of pathogenesis of infectious disease in 2013. In 2015, he won the Albert E. Levy Scientific Research Award at Emory, and then in 2021, he was awarded the Cavelli Fellowship from the National Academy of Sciences. So thank you, and we look forward to your talk.
Thank you. Thanks very much for the nice introduction. When I hear how long I've been here, I'm always like, what? Who are they talking about? Okay, so yeah, thanks for inviting me. And I'm going to tell everyone today about this type of antibiotic resistance that we study, heteroresistance, and hopefully can lay out for you why we think it's important, what it means clinically, and ways that we also can exploit it. Okay, so for anyone who doesn't already know, antibiotic resistance is a huge problem. And this says that it shows that by one prediction, that by the year 2050, there will be 10 million worldwide deaths annually due to antibiotic resistance, which is predicted to outpace cancer by that time. And already now there are over 1.2 million deaths per year globally. That is more than HIV and malaria combined. So this is a huge problem. So we started some years ago the Emory Antibiotic Resistance Center to try to bring all the amazing resources that we have here really together. And that completely has led to this project. So the goal was to bring the clinical microbiologists and clinicians and basic scientists and epidemiologists all together interacting. And that is what led to the isolate that I'm going to show you. So first, I'm just going to talk about, so there can be heteroresistance to many drugs. I'm just showing some data on colistin because it's, I think, the easiest example to see and it's really clear. So this is an e-test. There's a gradient of high drug concentration here down to low concentration here. And you can see this lawn of bacteria growing. And then there's this big zone of clearing that correlates with the drug concentration. So this is a susceptible strain with a big zone of clearing. This is what a resistant strain would look like. And you see a much smaller zone of clearing. So more bacteria can grow. And then this is the isolate that started off all of this. So it has the zone of clearing similar to a susceptible strain, but then it has all these breakthrough colonies. And this is indicative of likely heteroresistance. And so I really didn't know clearly what heteroresistance was when this isolate popped up in the clinical lab. But it really shows that how, any isolate that pops up, if people capture it and people study it with a weird phenotype, it can lead to who knows what. And that's what happened here. So huge thank you to Eileen Bird, who's the head of the clinical lab, for just like flagging this and letting us know. And then we dove in and tried to figure out what was going on here. So this is a population analysis profile. It's like the gold standard for studying heteroresistance. So we plate the different strains on increasing concentrations of drug, and then we score the percent surviving bacteria. So it's tons of dilutions of plating, but it's actually pretty simple. So a susceptible strain, see here, is all uniformly killed at a low concentration of drug. A resistant strain is killed at a much higher concentration of drug, but again, importantly, it's uniform killing. So the population is behaving the same way. All the cells are behaving basically the same way. This heteroresistant strain, which we call RS, because it had both resistant and susceptible cells, you see 90% of the cells are killed at this low concentration, so they're super susceptible. But then the subpopulation that remains is crazy resistant. Up to 500 micrograms per mil colistin, it's still surviving untouched. So that's like over 1,000-fold difference in the MIC between the susceptible and resistant cells. So if we look at what happens when we treat in over time, you see initial killing of the overall population and then outgrowth. And you can see here that this initial killing is down to the level of the pre-existing resistant subpopulation. So all the susceptible are killed off and the resistant are untouched and remain. And then over time, the resistant ones grow out as long as drug is present. And so this highlights that this is not persistence. There's something called persisters. These are like metabolically dormant cells that can resist drug because they're not doing much, basically. And most drugs need the cells to be actively trying to grow in order to kill them. So this is not that phenomenon because these resistant cells are growing even when the drug is there. And so here again, I'm showing pre-treatment level of resistant cells. What happens when we treat with drug? We select for the resistant cells and greatly enrich for them. Now, when we passage from here subsequently without drug, if this were a resistant mutant that had developed and got enriched, this would not go down. But we see that this in one subculture reverts to the baseline frequency of the resistant cells. So it's super flexible and dynamic. And this ability, this is part of the reason that this, I'll show you, is really hard to detect, because for some of these drugs in one passage, as what happens in the clinical lab, then you don't see the resistant population in some cases. So we didn't know at this time if this heteroresistance mattered in an in vivo system. Here we're showing a susceptible strain. We give a really high inoculum, so all the mice succumb to infection. But then when we treat with colistin, we can rescue all the mice. In contrast, when we take the heteroresistant strain, we see that all the mice succumb to infection. So this resistant subpopulation is mediating a treatment failure in an in vivo model. And then we looked around, like at first we thought, we didn't know if this was like a weird unicorn strain, that there were no other strains like this. Unfortunately, there's tons of them, and this is a common phenomenon, but we found this was one of the next ones that we found, that it had, it's sort of the same idea, but it has a much lower frequency of the resistant cells. So like 1 in 10,000, 1 in 100,000. And it also led to treatment failure. So even though treatment with the drug should be causing 99.999% of these cells to be killed, we're still seeing treatment failure. And then what was really scary was this is what that strain looks like on the e-test. So it looks completely susceptible because the frequency of the resistant cells is so low that they don't show up here in the zone of clearing. And this is classified in the clinical lab as susceptible. So these strains that we're totally missing where the clinician would think it's safe to treat can nonetheless, in a mouse model at least, like cause treatment failure. And I should have said at the beginning, all of these isolates I've shown so far are Enterobacter, so related to E. coli, but they're a little bit different. So they're Enterobacter. So I haven't presented this for a long time, but I wanted to go into more of the broad biology of this during infection. So this is just showing the percent of the resistant cells from the infection when we treat with colistin. And so we see this increase in the resistant cells. What was surprising to us, this was initially just a control, but then really interesting that we infect and don't treat with colistin, and we're still seeing increase of the resistant frequency. So what is driving this? And I should highlight, this isn't going to be true necessarily for every drug, but it is for some. So we started to try to think like in a mouse without drug present, what pressure would be on these bacteria. And so we thought the immune system and macrophages. So this is the pre-infection frequency of resistant cells. This is if we infect, we see this increase. And then if we deplete macrophages with these claudrinate liposomes and PBS as a control, we saw that we didn't at early time points after infection get enrichment for the resistant cells, suggesting that the macrophages are driving this. And then so we looked at individual antimicrobials in vitro. And so one important thing to note is that colistin is highly cationic and antimicrobial peptide. And so lysozyme is highly cationic as well, and antimicrobial enzyme. And then cramp is the murine ortholog of L37, which is a human cationic antimicrobial peptide. And we see that with increasing concentrations of these, we can select for the resistant cells. Then this was meant to be a control for these, but then even the hydrogen peroxide also was able to select for the resistant cells. So it's indicating these colistin resistant cells are resistant to much more than just colistin. And then here again, we took wild type mice and we see this selection. When we made these triple knockout mice that were lacking all of the antimicrobials that I just showed you, like their lysozyme knockout, cramp knockout, and the NADPH oxidase knockout, which generates hydrogen peroxide. And then you see that at these early times, you don't get selection. So it's specific antimicrobials from our host immune system that is driving the increase in the frequency of the resistant cells, even before drug is present. And so this is worrisome because if you have this situation before you even give colistin, then what's the chance that colistin is going to work? And so we think there's just this like molecular mechanism crossover in resistance between these and colistin, which is driving this. Okay, so how frequent is heteroresistance to other drugs? And so this is one of the things that we're so lucky to have here at Emory. So this great surveillance initiative, so MUGSI from the Georgia Emerging Infections Program. So we're looking at 104 carbapenem resistant Enterobacteriales. And so the first thing I would point out here is that, so this is in this circle, we're showing how the clinical lab classified these. So everything in black is resistant and everything in gray is susceptible. So you can see these are pretty resistant strains. They're carbapenem resistant. They have a lot of other resistances. And then in blue here, we're showing what percentage of the times that a strain was tested against a given drug was it actually heteroresistant. So we see, and in other studies, we see that heteroresistance as a phenotype, as a mechanistically is present about 25% of the time. So this is very prominent. And then here we're just taking all of the strains and all of the antibiotics that we looked at and saying the ones that were classified resistant by the clinic and our approach are the most resistant ones. The ones where we agree they're susceptible or very susceptible, but then there's two populations of isolates that we thought were heteroresistant to a given drug. And you can see here the ones that are classified resistant in the clinic versus the ones that are classified susceptible in the clinic. There's a difference in their frequency of resistant cells. And I'll show this again. So if you're heteroresistant to a given drug, but you have a high frequency of the resistant cells, you will be classified resistant. But if you have a low frequency of the resistant cells, you will come up susceptible. And that's what we saw previously with colistin, where one was seen to be resistant with the breakthrough colonies and one was not, and it looked susceptible. Okay, so we saw heteroresistance to every drug that we have looked at so far. Sometimes these are rare, like for ampicillin, where almost all 99% were resistant. But then for others, for like phosphomycin, heteroresistance is actually the norm phenotype. And I can also say that these were carbapenem resistant, so they were presumed to be resistant to meropenem, but half of them were actually heteroresistant rather than resistant, just with a higher frequency of resistance. So this is everywhere. We've focused a lot of effort on beta-lactams because they're really the most prominent class of most used antibiotics. So 65% of injectable antibiotics in the US in this decade, 2004 to 14, 47% of outpatient prescriptions in 2020. And then this is showing the development pipeline. You can see it is largely dominated by the blue that are beta-lactams or beta-lactamase inhibitors. And beta-lactamases are enzymes that degrade beta-lactams. And then all other classes are combined, are shown in gray here. So this is most of what we're doing right now in the pipeline is making cousins of beta-lactams that have already been developed. And that's a problem that many people think, as you'll see. So cefiderocol is one of these, is the newest beta-lactam. And it's a cool drug in that it's a cephalosporin, but linked to a siderophore. So siderophores are ways that are molecules that bacteria use to eat iron, to bind iron and take it in. So this was a super cool idea. It's like, let's link a drug to the siderophore. When the bug tries to eat iron, it's going to end up taking up our drug, and then it won't be resistant. It won't be able to like turn off the iron eating, and so it won't be able to prevent this from getting it. That's super cool. When I heard about it, I was so jealous that I didn't think of that. And unfortunately, it didn't quite pan out so bugs are clever, and there's lots of resistance mechanisms to this even. So this drug is thought to be totally resistant to extended spectrum beta-lactamases and serine carbapenemases. Okay, so the company Shionogi that did that brought this drug to market did a phase 3 trial called Credible. And in red here, you can see the mortality rate in this phase 3 trial for different bacteria and then the total. And so you see this like scary high level of mortality. So this is a new drug that's now been FDA approved even after this trial. And 50% of patients with carbapenem-resistant Acinetobacter died after getting this drug. And it was not clear why this was happening because the in vitro data looked really good. So we haven't been able to get the isolates from the trial, but we assembled with the different surveillance networks that we have here, a collection from Georgia that was tried to approximate like the types of bugs that were in this trial. So importantly, these are from predominantly 2014, and I think this drug was approved in 2020. So way before the drug was even out. And the rate of heteroresistance is shown in black. So you can see that it sort of closely approximates like the mortality rate, which makes us think that potentially heteroresistance was the cause or part of the cause of the treatment failure. Another important thing to show here is if we use, this was from the trial, the rate of resistance that you detect using standard broth microdilution, where the heteroresistant strains do not necessarily grow out and are not seen. So basically the company is sitting here like 90-something% of these should be susceptible, but half the patients died, and what's the difference? And so we're thinking that potentially it's due to this undetected heteroresistance. Oops. So I stole these slides from a meeting from last week where like people were doing talks after this. So that's not going to happen, but it happened last week. Okay, so this is some data comparing different, I've alluded to this, but comparing different tests. So Here's again the mortality rate for the Acinetobacter and Enterobacterales. And you can see none of these tests, broth microdilution, agar dilution, disc diffusion, they really don't detect the resistance, but this population analysis profile does. So unfortunately, this is not something that could be used. It's too labor intensive for it to be used routinely in the clinical lab. But also highlighting this point of the difference in frequency, you can see that of the ones that were detected as being resistant by any of these standard tests, it correlates with both the Acinetobacter and Enterobacterales as the ones that have the higher frequency of resistant cells. So the ones in red have a higher frequency, and roughly at this line of 1 in 10 to the 4, if you have fewer 1 in 10 to the 4 fewer resistant cells, you're pretty much not going to be detected by these tests. Okay, so we dug in further to one of these strains in particular that was heteroresistant to Cefidrocol, and you can see the curve here where you have this resistant subpopulation sort of hanging on, in contrast to a susceptible strain where it's just dead. We found, and so many people have found now that gene amplification of resistance genes, like increased copy number in a given location in the genome. So you can think of it, it's sort of like that the genome is flexible and that you can get more copies through recombination and tandem repeats. And then this is flexible where you can get more or fewer copies. And so we found that this region here was amplified in these resistant cells, and it encodes the beta-lactamase SHIT5, which is an ESBL. So like, long story short, this is a pretty wimpy enzyme that should be crushed by Cefidrocol and should not be able to provide resistance to Cefidrocol, but then we're seeing these resistant cells. And so what you can see here, what's going on is that here's again the path where you see it at higher concentration, fewer cells surviving. If we look at the copy number of that region, it's inversely proportional to the number of cells surviving. And so if we go through a couple of these, so at 8 micrograms per mil, there's only 1 in 5,000 cells that's resistant. And on average, they have 2 copies of this region. If you look at 32, it drops to 1 in 150,000 resistant cells, but they have on average 20 copies of this region. So this increased gene dosage essentially is a huge problem for beta-lactams and other drugs. And that's how you can have a strain with sort of wimpy activity, or amplifying something with a wimpy beta-lactamase, but then nonetheless promoting these cells to be resistant. And this strain, if we measure the MIC by, again, the standard test, the broth microdilution, it comes up very susceptible. So 16 is the resistance breakpoint. This is way below that. But if we expose it to Cefidrocol one time, it's above 64. So these, you can see the resistant cells are there. This is just showing that in a sort of in vivo model, so these are worm larvae, that you can see that if we treat with Cefidrocol after infection, we can enrich the resistant cells. So like this happens in an in vivo system. And then also we drive an increase in the copy number. And then this is a different isolate. This one is Klebsiella, but this was from a clinical case at Emory, where the patient, so the disc diffusion classified this isolate as susceptible. It had actually a really nasty beta-lactamase, NDM5, and Cefidrocol therapy failed. We saw when we looked at that isolate after the fact that it was heteroresistant, that we found this region with NDM, including NDM5, and we can see in vitro in the lab, when we treat with colistin, we get amplification of this region. So this doesn't prove that that's what happened in the patient, but it's correlative at least. So we are concerned about this. And then I think the next data really explain like why is this potentially a problem beyond ceftriaxone and much bigger than this and potentially threatens the pipeline is, and so here what we're showing is, so first of all, if we knock out SHIV5, which is shown here, you can see you lose the resistance cells. So resistance is considered at 16 and these drop down when you knock out that gene. So it is dependent on that gene. If we use clavulanate, which is a beta-lactamase inhibitor that can inhibit SHIV5, we can see that we also become susceptible. So combining these beta-lactamase inhibitors is very effective, and that's why so many of these are in development. So this looks great. That being said, if we go to a slightly low amount of ceftriaxone 8, and then we put in tiny amounts of clavulanate, you can see that there is this correlation where fewer and fewer cells are surviving as we go up with clavulanate, but those that survive are increasing their copy number. And so we are concerned about that because it shows that at least there's a way for these cells, resistant cells, to be able to survive, even if at this point they're not up to the level of resistance that we would worry about in the clinic, let's say. But here we're showing, so we start off with the strain that's susceptible, and then we have clavulanate present, but then we start increasing the concentration of drug in sequential passage. And you can see that the curve is the resistant cells. Well, the cells that survive are becoming more resistant going this way, and also the frequency of them is increasing. And so you can see that here, if we look at the copy number during these passages, you can see that the copy number is increasing as we increase the stringency or the stress on these with higher drug and higher beta-lactamase inhibitor. And so I think this really nails like a really critical point for this part of the talk. For the strain to survive at 16, it needs 6 copies of this region, including the SHIV5 beta-lactamase. For it to survive at 16 and with clavulanate, now this is the same number of cells surviving, but they need 87 copies of this region to be able to survive. So that's great in one sense, that clearly the clavulanate is having an effect. It's making it much harder for them to survive, but these bugs, which should be completely eradicated by ceftriaxone and clavulanate together, they do have a pathway to survive and be resistant if they have enough time to get enough high enough copy number. And this further drives home that point. So there's we made a point mutation that's known in the literature to weaken significantly the activity of SHIV5. And you can see here that even though this is like, so SHIV5 I said is weak, and this is like a sickly SHIV5 essentially. You can still over time get survival of this mutant, and you can see that you can increase copy number up to 150 copies of this region, which brings up some really interesting questions about like, what does this mean for the size of the genome and the fitness costs? But when it's either get killed or survive, this is better than getting killed. And again here, this is just showing more strains that we looked at from different types of bacteria that were all considered by standard testing by broth microdilution to be susceptible. And then we passage with ceftriaxone and you see this resistance jumps out. So this is not restricted to Enterobacter. It's present in many different bugs that are relevant in the clinic. And so just to sort of summarize this part, the ceftriaxone heteroresistance. Oh, and I guess I didn't actually say this on that slide. So when you see survival of strains at these different concentrations, like fewer and fewer as you go higher with drug, it's really indicating that there's a continuum of resistant subpopulations. And what we had thought, like in the first example I showed you with colistin, it was susceptible cells and resistant cells. And there were two populations. The resistant ones were super resistant, the susceptible were very susceptible. That was it. It was binary, basically. Here, what we're seeing is that this gene dosage is creating a continuum of resistant cells with increasing constant or resistance to the antibiotic. And so we didn't realize that existed in that way, and that it was so much more complex and heterogeneous than we thought. Okay, so the really important thing is here that the beta-lactamases with weak activity that can mediate resistance when they're at a high enough copy number, a high enough expression level. And broth microelution and other standard ASTs don't reliably detect heteroresistance. So what does this mean for the pipeline? And this is how we see it. So if you look at how drugs are currently developed, especially beta-lactam, beta-lactamase inhibitors, the first step is an enzymatic assay. So you take a certain amount of beta-lactamase, which is the main known resistance mechanism, and you take a huge library and you find compounds that are degraded by this thing, and then they're not effective, or you find some that are resistant. And the ones that are resistant to the beta-lactamase in vitro move forward in the process. So this does not necessarily take into account the fact that you can have 150 copies of the beta-lactamase. So they get to a point where they say, you know, this compound, this new compound is 50 times more resistant to a given beta-lactamase than our predecessor drug, and we're going to move forward with that. And that sounds great, except if the strain can become 100 times more resistant, then being 50 times better isn't going to be sufficient. So then, unfortunately, at the next step, they screen for, let's go out of biochemical assays and let's go into taking strains from real clinical isolates and saying, does the drug work or does it not work? So, because this is more in the sense like real world resistance, at least it's in whole bacteria. But unfortunately, if the frequency of the resistant cells is 1 in 100,000 or something very low, it looks like broth microdilution works. You're only detecting the ones right here that have pretty overt resistance. And so drugs move ahead. And this is because in broth microdilution, there's not enough time for the resistant cells to grow out and take over the population to where the whole media becomes cloudy. And so they're not seeing that this population is there. And then, actually, I can highlight there's an in-between step. They go into animal models, but then again, they get duped because they take their time point is usually 24 hours. So at 24 hours, you have the drug looks good. You're still having killing of the susceptible cells, and there's only few resistant cells. So they stop, they look at the animal 24 hours, the bug load is going down. They're happy. They don't realize that by day 3, it would be expanding and increasing. So they don't see that. And then finally, they're in people now, giving this to real people. And you get to this clinical trial, like what I showed you, and over 7 or 14 days of treatment, these resistant cells that are very few take over, they expand, they amplify their genes, and now they cause treatment failure, and nobody understands why this treatment failure is happening. So in no way am I saying that heteroresistance is like the main cause of these treatment failures or the only one, but it seems so far to be a prominent cause of these otherwise unexplained treatment failures. Okay, so I guess I'll just keep going. If anyone has like a burning question about this part, feel free to interrupt. The next part is like happier and is a bit like, how can we potentially exploit this phenomenon to actually treat the most resistant strains, which was also really unexpected. So from those 104 carbapenem resistant isolates that I showed you, we looked at how many are resistant or heteroresistant to multiple drugs, and the vast majority here, almost 90%, are heteroresistant to at least 2 drugs. So that will become interesting. So then I'm going to show data for one isolate in particular that is heteroresistant to 3 drugs. What's important here is that they're from 3 different antibiotic classes. So colistin it's really in its own class, basically. Ceftazidime is a beta-lactam, and fosfomycin is in its own class. You can see here that the path curves for this isolate on the different drug, they're all different. So again, for colistin, you have like this binary thing, and then here it's more gradual, but even these differ from one another. And so that already suggested that maybe the resistant subpopulation for each drug was different, like different cells. But we didn't know that yet. So, and then this is just a control that like there is standard conventional resistance in this strain, like to ampicillin, this is just resistance, right? So it's not like it's heteroresistant to every drug you could ever throw at it. So this is the frequency of resistant cells for colistin. Then we genetically knocked out FOQ, which is a regulator of colistin resistance. And you see that the colistin resistant cells are gone. So they depend on FOQ. However, when we look at the fosfomycin and ceftazidime resistant populations, they don't change. If we look at FOS-A, which is a resistance gene for fosfomycin, you see that this drop in resistant cells is specific to FOS-A and fosfomycin, and then the same thing for ceftazidime with AMPR. So we're making individual deletion mutants and affecting individual subpopulations, like really proving that these are distinct populations, which is just nuts to me. Like I guess it's hard to remember now, like, I don't know, 6 years ago when, we were doing some of this, that there are so many subpopulations that are different. And like, we know that these types of bugs can be, exhibit bistability in flagella expression and other traits. And so, like for me, when I was taught in grad school that like you pick a single colony and you grow it up in a tube overnight, and those cells are identical, aside for some very random, like 1 in 10 to the 8, mutants. Now I kind of come in lab and I see it too, and I wonder like if any 2 cells in there are exactly the same, exactly doing the same thing, because there's so many of these traits that exhibited by only subpopulations. So we were actually, this came out of actually doing an experiment to try to prove further that they were separate, these subpopulations. And so we combined drugs, and we're like, well, if they're separate, you should not have a subpopulation or very few that are resistant to both drugs combined at the same time. And so that's shown here, like for drug A is, let's say 1 in 100,000 resistant cells, and drug B is 1 in 1,000. If you combine those drugs, you should have an unbelievably few number of resistant cells. And so that is what we've tried to look at here. So we're taking just colistin and fosfomycin first. And this line is, if you just through math say that the killing would be additive, this is the curve that we would expect to get. And then we combine the 2 drugs, and that was basically the curve that we got. So, and we see the same thing for colistin, ceftazidime, and also ceftazidime fosfomycin. So this is not restricted to which combination you're choosing. And then very importantly, so I showed ampicillin does not kill at all. if you combine ampicillin with colistin, you don't see any additive killing beyond what colistin can do on its own, and the same for the other combinations. So we were excited about this. And then we did a big screen to look at this. So we took 8 clinical isolates, 16 antibiotics. So that's a total of 120 different combinations of antibiotics. And had nothing to do with this, but I know the lab was like just covered in plates for like a week or more. And so, okay, so this is, just let me know if you have a question with this part, because it's important to get your head around this. So for every combination, for every isolate, and for every 2-drug combination, the clinical lab would have called either the strain susceptible to both of those drugs, resistant to both, or resistant to one and susceptible to one, because they don't have a designation for heteroresistant. So we are going to now focus only on the ones where the clinic called it resistant to both drugs. And then we do our test for heteroresistance, and we're going to look at 2 different things. The ones that where our testing for heteroresistance agrees that this strain that classified resistant to both drugs actually is resistant to both, and where it was classified resistant to both, but it's actually heteroresistant to both. And so what we see is if we look at the ones where it really is resistant to both, we get almost no killing, so 0 logs of killing in the vast majority of them. But if we take the ones that they were thought to be resistant to both, but there's actually underlying heteroresistance, we see on average 4 logs of killing in those strains. And so I guess I just also want to highlight that this changes the model of, so there's this idea of drug synergy, like when a strain is resistant to 2 drugs, but you put them together, that there's synergy, and that indicates somehow that these 2 drugs, when hitting the same cell at the same time, like work in concert to promote killing. And what this model suggests is that happens very infrequently. That's what's shown here. What our model is suggesting is that you don't need the 2 drugs to target the same cell. You just need each drug to kill the cells susceptible to it, and then you end up with this like additive killing. So it changes how we look at drug synergy. And so there was this case of a woman who went to India and broke her leg and went to the hospital and got this thought picked up there, this super resistant, pan-resistant Klebsiella, came back. The CDC, unfortunately, she succumbed to infection. They treated with like 4 drugs at the same time and couldn't find something that would help her. This is a schematic of some of the antibiogram, and you can see all the R's. And then we looked at that isolate using PAP, and we found instances of heteroresistance. So if we take amikacin, which is one of these, and piptazo, and we combine them, so you see it's heteroresistant to both. And then if we combine them, we can eradicate that strain as predicted. In addition, if you show over time, you see the bounce back after initial killing, the resistant cells grow back, same thing for amikacin. But if you combine these, we don't get outgrowth again of that strain. And you can even see just looking in an overnight, like the combo looks like the 2 that didn't have bacteria inoculated. And then if we do this in a mouse model, we infect with this strain, the single treatments can't rescue the mice, but the combo treatment does. So we were really excited about this as an approach to, I guess, make these combinations make sense, because just this is what clinicians are faced with in some of these terrible cases, which luckily don't happen that often here. They're essentially guessing like which R's to combine, and they don't really have, they're like, well, this works on a previous patient, this and this combination, so maybe I'll try that. But this is really isolate specific. So clinicians have clearly said, like, we're just guessing at this point. So there was a real clinical case at the Children's Hospital. So a kidney transplant patient with a urinary tract infection with pan-resistant Klebsiella. And so they were worried that this patient could lose their grafted kidney or get sepsis. And this is what the antibiogram looked like. So this is just like a disaster, especially for the carbapenems, where this strain had 2 carbapenomases, including NDM, which is like sort of the worst one you can have to some extent. So the clinical lab at Showa said, you know, if you want to test it, like they had heard about our approach and our paper, so they're like, yeah, give it a shot. Like whatever you do, don't bother testing beta-lactams because of these, like there's just no chance that this thing is susceptible in any way to a beta-lactam. So we saw, of course, because that's how things work, that it was actually heteroresistant to meropenem, which is still really interesting to figure out how that works, and to colistin, so to these 2. So the patient was initially treated with tigecycline, because it was like borderline susceptible to that, but that doesn't really work well in the urinary tract, so that didn't work. The infection persisted. And then they started this combo treatment, and after a very short time, the patient's urine cultures were negative. And so we think in the future that this is currently what we're looking at, what clinicians are seeing in an antibiogram. In these cases where there's a lot of susceptibility, you would want to know which susceptibles are actually heteroresistant, so you could avoid those. And for monotherapy, use the ones that are like true susceptibles. And that hopefully will increase the efficacy of monotherapy. In these awful cases where there's pan resistance, you want to know which of these you can potentially combine logically in order to still have salvage some activity. And then also I think in the future, like with future diagnostics, hopefully we'll even have resolution of what's the frequency of the resistance. It's like in this example, you would clearly want to combine meropenem and colistin, not use tetracycline, where you're going to get much less bang for your buck if 90% of them, or if 90% are susceptible, but 10% already are resistant. And that is it. So I want to thank all the members of our lab. This excludes a bunch of people who did some of the earlier work, and especially on the combo stuff that I just showed. And so that's like Victor Van and David Hupnagel, and then the people who really did the most for the stuff I showed before that are Jake and Tuba and Carter. And of course, as I said, Eileen, who, like, I think it's pretty cool that, you know, something can come, like, I'm not smart enough to know what to work on, but when an isolate like pops up in front of us and it looks weird, and if you just roll with it, it can take you to all these interesting things that we never would have worked on at all otherwise. So I think that's really the strength of what our antibiotic resistance center is doing. And then people in the EIP for all the surveillance isolates and funding. And that's it. With that, I'm happy to take some questions. Thank you.
Any phenotypes that are associated with like fitness cost with this resistance?
Yeah, so it's a good question. So Dan Anderson, he's in Sweden. He does a lot of work on heteroresistance. He has a paper where looking at the cost of different amplifications. And so it's a really interesting question, like, does the size of the amplified region, if it's weighted, because these can be up to like 250 kb, they can also be like 3 kb. So does that impact like how the fitness costs? So he has a paper showing that definitely there is some fitness cost. I don't think it's to the point of like measuring exactly at that level of resolution yet, but yes, and then for Colistin has perhaps like the highest fitness cost of resistance to it of any drug, probably. Because they have to really remodel their outer membrane in order to be resistant. They change the whole charge of their surface. So for that one, we definitely see where, yeah, that is also in part why we think the resistant cells revert to in the frequency of the population so quickly in one overnight. They go back to their like homeostatic level of resistant cells because they don't like being in a resistant state unless they're being selected that they have to be. So yeah, I think that's going to be so much more has to be done on that to really figure out like different dynamics controlling this phenomenon. Yeah.
2 questions. So you mentioned gene dosing as one mechanism for heteroresistance. Are you finding kind of different mechanisms for different antibiotics and different bugs? This is the first question. And then, as you mentioned, PAP is super labor intensive. Is there a strategy for people in resource limited settings to do something like that?
Sorry, so maybe the second question first. I mean, I guess I would say in resource non-limiting countries like here, we don't have an approach to detect heteroresistance. So, yeah, I guess what I'm saying is it's not like I think in our resource limited countries where possibly like labor and stuff is cheaper, they actually have a better shot at doing PAP than you would ever have here in a clinical lab here. But yeah, we need better diagnostics, obviously. And so like we're working on that. Some other people are working on that. But it's really a challenging problem because you sort of have to detect a needle in a haystack. And then you don't want to wait 2 days for this thing to grow out, right? Because there's a big push for like more rapid antibiotic. So it's like a lot of opposing forces on this one problem. And then you also can't get, I mean, if you need 10 million cells to detect like 1 in a million reliably, you need a certain load of bugs. So that's going to limit your ability to like take something straight from a patient and be able to do that testing, which is what a lot of people are pushing for, these really rapid tests. So I think these super rapid tests are going to have a really important place in the clinical micro lab, but they're not going to detect this phenomenon. So we're going to need like a second really sensitive test that can do this. Then as far as your first question, great question. There is so much heterogeneity in the heterogeneity. So like different isolates of the same bug are going to, you can't predict when you have an isolate, it's going to be heteroresistant to this, resistant to that. And so even from the same like surveillance system, we see a lot of heterogeneity. And then for Call, I don't think out of maybe like 7 strains or 6 strains or something that we've seen where we see an amplification, I don't think we've seen the same gene beta-lactamase twice that's amplified. So that's across some different types of bugs. So maybe you would think in different types of bugs, but in 2 Acinetobacter, I think they're different slightly. And so it's hard at this point to like look at a genome and say, oh, it's probably going to be heteroresistant to X. So yeah, and there are different mechanisms, like I didn't talk about it, but we think for some other drugs, there's metabolic almost heteroresistance, where it's not just this enzyme only, it's also providing, it's like the metabolic state impacts that. And so it's very diverse and super interesting, but like challenging to get to the point where you can like make predictions.
There's a couple of online questions.
Can everyone see that? Do you know what it is about some antibiotics that have very high heteroresistance rate versus very low, even within the same class? Great question. I think So the ones that are newer, that have been optimized for activity, there's probably gonna be more susceptibility. Then some that are super old, like ampicillin, the resistance actually is much more prevalent than heteroresistance. So in part, it's like the activity level of the drug when it was developed, or also how much it was used. So colistin is not used very often, and there's a high fitness cost, as we talked about. So the heteroresistance rate is like pretty low. So I think there are a lot of factors that can play into that. And then are there ways that drug development process could be improved by earlier awareness, be incorporated better? Yes. Thanks. So yeah, Julia, I think like for sure, the main way is that if companies were open to this even being an issue, I think that would be the first step. Like if it's a 12-step process or something, right? Like step one, admit your problem or something like that. I don't know. So, but then I think in practice, like they really need, I don't know how much they're gonna do without someone going to them and saying like, here's a new diagnostic approach that you can incorporate into your initial screening that will allow you to catch the heteroresistance. I don't know what necessarily if a drug company wants to do PAPs. Step 13, sure, yeah, look for heteroresistance. Yeah, exactly. So I think unfortunately there's a lot that has to change to get. The good news is like a lot of, we're talking about really resistant bacteria here. There was like the average strain from the average infection is going to be susceptible to enough drugs that it should be treatable still. It's just that we're moving in a direction where resistant there, the bugs are winning. And then also with this like predominance of beta-lactam cousins in the pipeline, it's like when I don't see when is the tide gonna turn if we're like most of our eggs are in a basket that is only gonna work for a limited time. So there's a lot to do. So if anyone's interested, we're always hiring. Come work with us. It's fun. You won't run out of questions because there's a lot to figure out here. So awesome. Thanks so much.
Thank you.